Hello, hello. Okay, now that I turned the mic on, welcome everybody. Thanks for coming. Today we have a very special seminar today to see what research the Skaggs Fellowship awardees have done during their time period. So the Skaggs Fellowships are the first full endowed fellowships that the college has been able to offer, and it's because of the generosity of the Skaggs family. This endowment is from the Skaggs Institute of Research. So this is the 6th year we've been able to offer these fellowships, and they were first awarded starting in 2016. And so on behalf of the college, we're so grateful for the Skaggs family and for these fellowships that further develop our student scientists and progress our research mission. And welcome to the donor rep, Ronnie here, who's come to see these talks. And I look forward to hearing the 4 student speakers give their exciting research presentations. So thanks everyone for coming. Still on.
All right, so it's my pleasure to introduce our first speaker for today's symposium, Ms. Stephanie Hurd, or I think as majority of the audience knows her as Billy. Billy received a dual BA degree from Kalamazoo College in Michigan. The first BA was in chemistry with honors. The second was in French, and she also did an internship in Strasbourg. So she's fluent in French. She also speaks Spanish and knows basic Japanese, which helped because one of her projects, she had to interpret a patent that was in Japanese. So very, very talented. In addition to being a tremendous researcher involved in many projects, she also is really involved in service here at the university and in Salt Lake community. For the university, she's been involved with the bioscience PhD program, where she's been lead chair, retreat chair, student representative for the Department of Medicinal Chemistry, and then also actively involved with LGBTQ and BLM activities and outreach in Salt Lake. Her project today that she's going to be talking about is multidisciplinary. Do you want to put down? Got it. Sorry. Is multidisciplinary, drawing from areas of biochemistry, bioinformatics, fungal genetics, and natural product structure elucidation. And so before I turn the floor over to her, I also want to give a warm welcome to her parents who were driving from Seattle and just happened to be passing through Salt Lake. So welcome. And with that, Billy, the floor is yours.
All right, can everyone hear me okay? Awesome. Thank you for the introduction, Jackie. So today I'll be telling you a few brief stories about how nature is able to create unique, clinically relevant small molecules and how we as researchers can exploit these strategies for the discovery of new drug scaffolds. Okay. So this is a comprehensive look at every FDA-approved small molecule drug from January 1981 to September 2019. And as you can see in yellow, about 1/3 of them are from traditional synthetic chemistry, but all other drugs in this plot are either in part or in whole derived from nature. In our current age of increasing drug resistance that we see in the clinic, we need new therapeutic strategies, and nature has continued to be a prolific resource of drug leads. So what are some of the molecules we find in nature? Here is just a handful of medicinal natural products, and these represent a wide variety of therapeutic applications. So we have some common clinical agents you may recognize, including antibacterials like vancomycin and tetracycline. We also have the antifungal echinocandin B and the antiparasitic artemisinin. But also included here is the painkiller morphine, the cholesterol-lowering agent lovastatin, the immunosuppressive cyclosporin A, and the cytotoxic chemotherapeutic paclitaxel, and of course, everyone's favorite and most acceptable stimulant, caffeine. So where do these molecules come from, and how do we find new drugs from these sources? All of these molecules come from plants, bacteria, and fungi that you find out in the world. These examples specifically don't include any animal-derived compounds, but those are also an important source. And each of these was originally isolated from its producing organism before then synthetic or semi-synthetic methods were used to reach the same final compound for commercial use. And though we use these compounds clinically, the individual producing organisms actually get some kind of fitness benefit in their environmental niches, and that's why they produce these compounds. So how are they made? Natural products are biosynthesized by a dedicated suite of genes that in bacteria, fungi, and some plants have been shown to generally remain clustered together within a chromosome. Here I'm using lovastatin as an example, which was originally made by the fungal producer Aspergillus terreus, and now it's a blockbuster anti-cholesterol drug. And you can see how fairly simple primary metabolites like malonyl-CoA can become unique bioactive structures through the activity of very specific enzymes in red, such as polyketide synthases. Another important thing to note when looking at this gene cluster I'm showing here is that these can often also indicate the molecular target of the encoded metabolite. So lovastatin, it actually inhibits the enzyme HMG-CoA reductase, which is an essential step in cholesterol biosynthesis in mammals. And what that's doing there is this is a duplicated copy of a gene from primary metabolism, which is required for survival. So in humans, it's lowering our cholesterol, but ecologically in fungi, this compound inhibits the same enzyme en route to ergosterol, which is an essential component in fungal cell membranes. And so thereby, it is almost like an antifungal compound. So these duplicate copies contain very specific mutations that allow for the producing strain to avoid its own metabolite's mechanism of action, and this is a well-documented survival strategy across producing organisms. Also, the location of this resistance gene in this cluster is not a coincidence. Because the original copy is sensitive to this metabolite, it makes sense for this resistance copy to be included near or within this cluster to allow for co-regulation of all of these genes at the same time. So when the compound is produced, the resistant gene is present to allow for survival. So that brings me to the 3 stories I'll be talking to you about today. They're very brief. I'll start with the cytocalicins. I'll move on to a more complicated example with penilumamide A, and then I'll end today by discussing Pestalone, which is a new project in the lab that actually has some exciting results. So the first fungal strain I ever worked on in the winter lab was Aspergillus flavipes CNL338. And we were able to acquire this strain from Professor William Fennicle at the Scripps Institution of Oceanography at UCSD. We sequenced and assembled the genome, and through genome mining, we were able to identify about 50 putative biosynthetic clusters. But the problem with traditional bioactivity-guided fractionation for compound elucidation from this strain is that it produces, like many other aspergilli, this suite of cytocalicins, which are very cytotoxic. So these are major components in any crude extract of this strain, and they tend to drown out the presence of other lower abundance metabolites of interest. So in order to make this strain a little easier to work with, we took this putative cluster we identified, and I worked on a gene inactivation experiment to correlate that cluster to that suite of compounds. This is a quick schematic of the workflow I used. Essentially, I used fusion PCR to generate a gene inactivation cassette, which upon transformation into fungal protoplast should integrate into the genome. Preparation and transformation of fungal protoplast is a very involved procedure, but just to summarize it, you have to harvest spores, grow them overnight into young germlings, and then digest their hyphae with a mixture of enzymes to generate protoplasts, which are just lacking their cell wall. And then through this workflow, you can do a PEG-mediated transformation of that that inactivation cassette. And then when you plate unselective media, you should see colonies that have integrated that into your genome. I can then replica-plate those colonies and then verify them. The first verification step I perform is genomic DNA PCR to confirm not only presence, but also the specific location of that cassette. And then I can also take extracts from the mutant and the wild type strains and look for specific loss of production of a variety of compounds. This is pretty traditional natural product discovery and characterization. But beyond confirming the cluster's involvement in this biosynthesis of those compounds, we wanted to look at the specific enzymatic machinery involved. So specifically, FFSA is the core biosynthetic enzyme of this pathway. It's a hybrid polyketide synthase non-ribosomal peptide synthetase, or PKS-NRPS. There's going to be a lot of acronyms, I'm sorry. So these are large megasynthase enzymes that contain multiple independently active protein domains, which I'm representing with those red and blue blocks right there. And each domain is responsible for the elongation and modification of a single building block into the linear chain. In blue, I'm highlighting the adenylation domain, which is responsible for the final incorporation of the proteogenic amino acid leucine. And this is an important step as it distinguishes certain classes of cytokalicins from others. So you can see here that these enzymes work together to make take very simple primary metabolites, build very complex structures with very specific activities. And we wanted to look at the specific substrate loading capabilities of that adenylation domain and how it's able to selectively activate leucine. So the non-ribosomal peptide synthetase substrate binding code is very well established in bacteria, which are more characterized than fungi. And it's essentially 10 amino acid residues that line the binding pocket of the adenylation domain. I'm only showing you 9 here because the 10th is actually behind the substrate from this perspective. And these residues create very specific interactions that allow for only certain substrates to enter that pocket and be activated and incorporated into your metabolite. So through our work with FFSA, we were able to add to the known fungal adenylation proteogenic building block code, and that's what I'm showing here. And this code can also allow us to use bioinformatics to predict the substrate loading of any domain in this pathway and in a variety of other pathways. So this is a really powerful tool, and we were able to add to that knowledge. But what happens when your building block is unique, highly modified, or non-proteogenic? So that brings me to penilumamide A. This is isolated, this small non-ribosomal tripeptide came from the same fungal strain that the cytokalicins were isolated from, that's CNL338. And immediately we noticed on the left-hand side, it contains a very unique and highly modified 1,3-dimethyl lumazine 6 carboxylic acid building block, as well as on the opposite side of the molecule, a modified anthranilic acid. So when we did the genome mining, we found this cluster. It looked like it was correlated with this compound through a lot of biosynthetic interpretations, and we wanted to verify that again by gene inactivation. This is the same workflow I did before, applied to a different pathway. I was able to create the inactivation cassette, integrate it into the genome, verify that, and then see loss of production of penilumamide A in the mutant strain. So that's correlated now, that cluster to that compound. But some of these building blocks are so unique, we wanted to biochemically characterize the NRPSs involved. I was able to reconstitute all 3 NRPSs in this pathway intron-free in yeast using overlapping gDNA and cDNA. They're then co-transformed with this linearized expression vector into a yeast strain that will reassemble it for us as a circular plasmid. And then I was able to express all 3 proteins from yeast from the strain Saccharomyces cerevisiae BJ5464NPGA. This is a special strain that contains a chromosomally integrated copy of the NPGA gene from Aspergillus nigilans. And that is a phosphopantotheniol transferase, which essentially means every PKS and NRPS I purify from this strain is purified in its active holo form. I can then use those enzymes in a variety of in vitro assays. So when we took PLMA, J, and K, we combined them in every possible combination, and we only saw the expected assay product when all 3 enzymes were present together in that top line. And this was great, it was very reassuring to see, but we wanted to dive even deeper, again, look at specific domains that activate these unique building blocks. So I was able to annotate the domain organization of each of these NRPSs, and from that I could dissect out the A domains, which are the substrate-specific activating domains, and I was able to express them as recombinant proteins in E. coli. With those soluble proteins, I did more biochemical characterization that I'll get into in a minute, but those of you who are familiar with NRPS biosynthesis will notice that I have 4 NRPS modules here as these ATC units, and I'm generating only a tripeptide. And this is usually a 1-to-1 ratio of modules to monomers. So something weird is going on already, and that was why it was so important to us to look at all 4 adenylation domains and each of their individual contributions to the final product. Adenylation involves the activation of an acyl substrate by covalent addition of AMP. This can then be taken up by an acceptor molecule for chain extension, for example, in NRPS biosynthesis. And the adenylation reaction can be monitored by conversion of inorganic pyrophosphate to inorganic phosphate, and then addition of a malachite green detection reagent allows for colorimetric measurement at 600 nanometers. I performed this assay in triplicate with all 4 A-domain proteins using the full panel of 20 proteogenic amino acid substrates, including the unique compounds or the unique monomers found in penilumamide A. I'll just be showing you a small set of relevant data today, but here were the clear pattern of results that we saw after these assays. So it appears that PLMA is specifically activating that pterine 6-carboxylic acid unique building block. PLMK has a clear preference for anthranilic acid. And interestingly, the 2-module containing PLMJ seems to be activating methionine in both modules to approximately equal extents. But again, we want to use bioinformatics to look at what specific residues are selecting each of these building blocks and how can we use that for future prediction. So here are just the 2 non-proteogenic activating domains. Anthranilic acid has been studied by other groups, and these residues actually match up really nicely with what they've reported. But this is the very first instance of a pterin or lumazine-based derivative being activated by an adenylation domain. So now other groups looking at NRPS pathways can use that code and use it to interpret their own genomes. And that's now been added to this wealth of knowledge we have to draw from. And of course, we also did this with the 2 PLMJ A domains, which both activated methionine, and now all of those are available or will be available once this is accepted. So all of this work mostly led to us finalizing our proposed model for penilumamide A biosynthesis. So the gray domains are inactive based on extensive bioinformatic analysis that I don't have time to get into today. And based on that, it really does answer the question of how do we have 4 modules making a tripeptide? So the first module of PLMJ, which has an inactive thiolation domain, no matter how much substrate that A domain activates, there's nothing to tether it to the megasynthase, and therefore that entire module can't participate in biosynthesis. So now we have 3 modules actively making 1 tripeptide. So everything I've told you about so far has been about like the utility of genomic data for natural product characterization, and that's great. But if we can predict 1,000 compounds, it's not helpful if we can never isolate them, especially for clinical application. So traditionally, natural product discovery was this bioactivity-guided fractionation of a single strain in an isolated culture to look at everything it was making. And as new leads got discovered and few new ones emerged, people became really suspicious that maybe nature isn't producing as much as we thought it could. But what we understand now is that genetically, microbes are capable of producing a lot more small molecules than we see in isolated culture. And the problem arises from how we treat them in the laboratory. The laboratory conditions of isolated, very controlled cultures doesn't accurately recapitulate what they encounter in their environmental niches. And there are a lot of strategies you can use to activate these clusters, and that's something I want to talk about now. Namely, environmental factors, as listed on the left here, have been a really popular method to modify what a compound or what a strain is making. But that also doesn't take into account the biological relevance of all of the complicated signaling pathways in the middle here that these microbes have developed over an evolutionary timescale while they live in these diverse and complex communities together. So in order to access the chemical weaponry available to us from compounds like Pestalone, it's important to use a more complex strategy. And that brings me to my final story. Pestalone is a chlorinated benzophenone and polyketide. It was originally only observed when Pestalocious species CNL365, which is a fungus, was co-cultured with the alpha proteobacteria CNJ328. Both of these strains are marine-derived, and though the antimicrobial target of Pestalone is unknown, it has striking activity against methicillin-resistant Staphylococcus aureus and vancomycin-resistant Enterococcus faecium. So this is a very potent antibiotic, only seen under very specific culture conditions. And we're really curious as to how this compound is going to influence future discovery efforts for new drug leads. Natural products have always been a major contributor, especially in the antibacterial and antifungal drug discovery arenas. I'm highlighting natural products here in red, and about 75% of all antibiotics have been identified from nature. So we're really interested in how can we use this Pestalone story as a case study to activate other strains, find new molecules. It's all about finding new things that can help us in the clinic. So we started by sequencing and assembling the genome of the strain CNL365, which is another one we got from Bill Fennicle at UCSD, and we identified this putative cluster. It contains a very canonical PES-I regulator, that's a zinc 2 cis-6 transcription factor, but we also identified another gene, PES-A, that we thought might be regulating its transcription. PES-A is a putative homoserine lactinase, which is able to enzymatically quench quorum-sensing molecules. Quorum sensing is the bacterial chemical communication that it uses to regulate in large communities its growth and gene expression by the secretion of these N-acyl homoserine lactone molecules. So in the context of Pestalone, there may be some mechanism by which our fungus is receiving a signal from a competitor and it's using that in its own transcription of this antibiotic. Quorum quenching enzymes are able to inactivate this homoserine lactone signal. As I'm showing here, it's a simple hydrolysis of the lactone ring, and either one of these molecules may directly or indirectly be binding to one of these regulators to transcribe the entire cluster. So the presence of that gene in this cluster, it may be indicative that the fungus is hijacking a common method of bacterial communication in order to regulate the production of its own metabolites. So when we began this project, we just recapitulated the co-culture. We're using a custom piece of glassware that we've engineered in the lab. It's two flasks that are open to each other, but they're separated by a membrane. So this allows for media and chemical components to be shared, but no physical cell contact. And that was important for the transcription analysis that we wanted to do. So across a variety of time points, we see Pestalone production peak at the 5th day of co-culture. And using those same time points, we extracted RNA, and we're able to see that transcription of both the PES-A regulator and the PES-K core PKS gene are responding in the same way and are reaching that maximum transcript level at day 5. And that correlates nicely with the compound production as well. But we wanted to make sure that this was actually due to the presence of a quorum sensor coming from that bacterium. So we did a different study where we added in an exogenous, commercially available quorum sensor. So the fungus by itself is at the top here, and at day 10, you can maybe see some trace amounts of Pestalone appearing. But if we look at the lower plot, this is clearly amplified when we add a quorum sensor, specifically in this case NHHL, to this culture. And again, if we look at transcripts, both that regulator and that PKS core gene, in the fungus alone, they're accumulating at day 10, and then this is with the regular spiking of quorum sensor at 0.5 micromolar from the day of inoculation, we see pretty consistent levels of both of those genes across our time course. So this was really exciting. It's still preliminary evidence, but we wanted to look more closely at this PES-A regulator because it might be doing something pretty unique that's not been characterized in fungi before. So recently, a postdoc in the lab, Dr. Peng Zhang, and a rotation student, Bryce Wallace, were able to make a PES-A gene fusion with a GFP signal tag. They put this into a plasmid and they transformed it into Aspergillus nigilans 1145, which is a model fungal strain we have in the lab. And this was an important step because we wanted to remove this regulator from its host hierarchy of gene regulation. Fungal gene regulation is really complicated. There's a lot of levels and different responses. And we wanted to take that out of that context and just study this regulator in a well-characterized and genetically tractable host. So we put it into this strain, and what we're currently working on is using the same conditions of induction as we previously have seen Pestalone production, but now our readout is the quantification of GFP fluorescence. So both with a challenger and with a quorum sensing molecule, and we can compare those to negative controls. We just started measuring these results this week, and it does look like we see an increase in GFP fluorescence with the presence of one of these two inducers, but we're still working on that. So stay tuned for more in-depth characterization of that regulator. And in the future, that could be used for a variety of synthetic biology applications to induce the transcription of any gene of interest in theory. So to conclude, natural products is a multidisciplinary and community-driven field, and each step along this way can really impact your downstream productivity. So not only can you take a strain of interest and culture it in a variety of conditions to isolate new natural products, but you can also sequence the genome to identify putative clusters and use a variety of genetic engineering approaches to correlate those clusters to those compounds. But even more importantly than that, you can use clusters to biochemically characterize the specific enzymes they produce to investigate the regulation of these key pathways, and you can compare your clusters to public databases to add to the bioinformatic capabilities of the field as a whole. And with that, I'd like to thank so many people for helping me along the way, namely a postdoc, Geng Wei, and our other postdoc, Dr. Peng Zhang, and then our rotation student, Bryce, and our fantastic high school student, Jeanette. And of course, my thesis committee for their support throughout this process, and our funding sources, especially the Skaggs Fellowship, which supported me for this past year. And I'll take any questions. Bailey.
inactivated P domain, that's modulated. For some reason, it's still maintained there, it's still able to ventilate thiamine. And I'm wondering if we could get sequences of like related strains, or possibly that's still active, product.
So, yeah, because it looks like a remnant. So the question is about the second story I shared with pennilutamide and that kind of redundant module that's still present in the pathway. When I did a lot of bioinformatics to try and figure out where there might be something off. And specifically with that module, I regularly came into small modifications in the sequence. I don't know if that's the result of, an inactive or unnecessary domain that's now slowly being mutated out of the sequence. But there were also dimodule NRPSs that it was similar to still. I think it gets into that gray area of like, these are giant enzymes and they're similar, quote unquote similar to a lot of things at the amino acid level. But when you pull out those binding domains, you start to see like, okay, this specific residue is mutated. I don't think it's active, or it's gonna lack some necessary function. So we found some things it was similar to, but yeah, that took a lot of bioinformatics to pull out. Yes?
What project was with Scripps?
Oh, so we have an ongoing collaboration with Dr. Bill Fenical at the Scripps Institution of Oceanography. He sends us a lot of strains, and some of these Jackie brought with her when she founded her lab here. So both of the fungal strains I talked about today, all 3 stories, came from strains from his lab. Definitely. I don't think anyone is looking at biosynthesis of that compound in any way. So we've got a lot of side projects with that are ongoing. We're looking at, how it's actually made enzymatically. But as far as I know, that level of quorum sensing regulation has been barely studied in fungi. In bacteria, quorum sensing is great. It's really well understood. But in fungi, we're always like 10 or 20 years behind the bacterial side of things. So they're just catching up, but it looks like it's going to be a really unique story. Yeah. Cool. Well, if there's no further questions, thank you.
Well, it's my pleasure to introduce the next speaker. Sorry, is that on? Okay. It's my pleasure to introduce the next speaker, Katie Burrell Gerbers, new addition to the name. I need to get used to that. Katie is a 5th-year student. She's been in my lab for the past 5 years or 5 years. Time flies, especially when you don't remember a couple of years with COVID. Anyway, she came to us through the biological chemistry program and the University of Arizona, and since joining lab, she's been working on a project that at the time was brand new. It's actually been a very challenging but really quite revealing and intriguing project, and she's made some fantastic progress along the way, trying to understand the physiological roles of the TRPV3 ion channel and airway epithelium, particularly in the context of injury and repair associated with exposures to ambient pollutants that oftentimes interact with and damage lung epithelial cells. Certainly opened the doors of opportunity for a number of new areas of research in the lab. In addition to the work that she's doing in the lab, she's been involved in a number of leadership positions, a graduate communication committee, I think it was, here at the university, as well as a couple of committees with the Society of Toxicology and regional chapter of Mountain West Society of Toxicology. She's received several awards for posters and presentations along the way, and I will let her explain the details of the project. Thank you.
Okay, thank you, Chris, for the nice introduction. So, as Chris mentioned, I will be talking about my project on transient receptor potential vanilloid 3 or TRPV3 and its potential role within the lung epithelium and its response to injury from wood smoke or particulate matter or other forms of lung injury. So first, I'll briefly go over an outline for this talk. I'll go over the relevant background information, our rationale, so what sort of led us to look at TRPV3 within the lung epithelium in terms of repair, a few of my hypotheses for this project, and then I've kind of split this talk into two parts where I'll first go over my in vitro work on TRPV3 and its role in wound repair in cultured human lung epithelial cells. And then for part two, I'll briefly go into some of the in vivo work that I've been working on with our TRPV3 knockout mice and naphthalene exposures as a form of injury in these mice. And then finally, I'll go over conclusions and future directions for this project. So wood smoke particulate matter is the environmental exposure that led us to look at TRPV3 within the lung epithelium. Obviously, it's bad for you to inhale. Long-term exposure can exacerbate certain diseases such as COPD and asthma, but it can also cause diseases such as bronchitis, pneumonia, asthma, emphysema, and even non-smoking patients, as well as premature death. The EPA has also estimated that breathing in wood smoke particulate matter, especially on bad air quality days such as this day a few months ago, August 6, 2021. So the EPA has estimated that inhaling this wood smoke particulate matter exposure is equivalent to smoking 4 to 16 cigarettes. However, the toxicological outcomes of wood smoke particulate matter, as well as how chronic exposure to particulate matter leads to these chronic diseases, isn't fully understood. So in the Riley lab, a lot of us are pulmonary toxicologists. We utilize different models of particulate matter to hopefully identify a link between these environmental exposures and the chronic diseases that come out of them. And some of the molecular sensors within the lung epithelium that we've identified to be activated by particulate matter include transient receptor potential channels or TRP channels. So these are a huge class of ion channels that are expressed throughout the body. The family tree for these TRP channels is shown on the top right. These are non-selectively permeable to divalent cations, including calcium, and they're expressed throughout the body. Earlier this month, the Nobel Prize in Physiology was awarded to two researchers who have done a lot of foundational work on these TRP channels as well as piezo channels. And those researchers are David Julius and Artem Patapoutian. They did a lot of work establishing TRP channels as sensory mediators for temperature, the sensation of temperature as well as touch. So like I mentioned, we've identified TRP channels to be activated by certain models of particulate matter. TRPV3, which is what my project is centered around, is activated by wood smoke particulate matter, including pine and mesquite wood smoke particulate matter. So this first graph on the left is showing a calcium assay in which we have different isoforms of the TRP superfamily overexpressed in HEC293 cells. And on the y-axis is calcium flux as a percentage of a positive control. And you can see that as compared to their own unique positive controls or agonists, both TRPA1 and TRPV3 are selectively activated by pine and mesquite wood smoke particulate matter. And then on the right-hand side, this is showing transcript levels of our two TRP channels that we study within the lab. I do have colleagues who have been trying to figure out the importance of TRPA1 within the lung epithelium, but for the remainder of the talk, I will only focus on TRPV3. But we do know that both of these channels are expressed in lung epithelial cells. So on the x-axis are all of the human airway epithelial cells that we use in the Riley lab. And you can see that TRPV3 is expressed in all of these cell lines. And so for my in vitro work, I have utilized these Bs2B cells, as well as HBEC3KT cells, which are both human bronchial epithelial cells that have been immortalized. So what we know about TRPV3 in the literature, TRPV3 is actually one of the lesser researched TRP channels, but we do know that it is the most highly expressed TRP channel within skin keratinocytes. And so there has been a lot of work on TRPV3 within the skin, and these studies have associated TRPV3 with proliferation, wound healing, differentiation, itch, and hair growth. And there exists a sort of theme that comes about when you read about TRPV3 within the literature, in that if you affect TRPV3 expression or activity either way, you kind of get these extreme phenotypes out of it. So I've included this scale picture to kind of demonstrate that fact, whereas on one hand, when you have a gain of function of TRPV3 activity, you can have extreme phenotypes such as Olmsted Syndrome. So this is a rare genetic disorder in which patients have a overactive form of TRPV3 due to a point mutation, and they tend to have thick keratonic plaques on their hands, feet, and face, so almost like an overgrowth of keratinocytes. And then researchers have also investigated TRPV3 within hair follicles by culturing human and mice hair follicles. And when they add a TRPV3 agonist to these cells, they've noticed that it results in complete hair loss. And then on the other side of things, with the loss of function of TRPV3, TRPV3 knockout mice tend to have curly hair and whiskers. So this deficiency in TRPV3 leads to altered morphology of hair. We've also noticed this in our own TRPV3 knockout animals. When they're first born, they tend to have really curly hair. Also in these TRPV3 knockout mice, researchers have demonstrated following tooth extraction, they have a severely impaired oral wound healing as well. And I've also included the schematic from this paper by Chang et al. It basically shows TRPV3 and calcium flux and its signaling partners. And this paper really detailed that TRPV3 signaling within skin keratinocytes associates with EGFR signaling, which will become important later when I talk about EGFR signaling within the lung epithelium. And so another piece of data that sort of promoted us to look at TRPV3 in terms of wound repair following wood smoke particulate matter exposure was this in vitro or in vivo work where we treated C57 black 6 mice subacutely via the orphangeal route every other day for 3 days with pine particulate matter. And then 24 hours post the final treatment, took the lungs for qPCR transcript analysis or histology to look at the morphology of the airways. And in A, this is showing a quantification of TRPV3 transcript levels in the pine particulate matter treated mice as compared to the saline control mice. You can see that TRPV3 has increased in expression significantly. And then furthermore, when looking at the airways, the saline control is fairly normal in morphology. But interestingly, with pine particulate matter treatment, and as highlighted with the red arrow, you can see a thicker layer of epithelial cells in the airways of these mice, indicative of keratinization of the airways. This also correlated with an increased level of airway resistance when we ran pulmonary mechanics on these mice. And then when we treated with a TRPV3 antagonist, so 007 is our selective TRPV3 antagonist, there weren't too many differences in the morphology of the airways of these animals, maybe a slight accumulation of epithelial cells in this histology slide. However, more interestingly, co-treatment of the TRPV3 antagonist with pine particulate matter, you no longer see that layer of epithelial cells. So TRPV3 is expressed within the airways of both humans and mice, and I've shown you that TRPV3 is activated by wood smoke particulate matter. And then from the literature, we know that TRPV3 activity and expression has been linked to wound repair within the skin, but also within the mouth and eye epithelium. So taking these pieces of information, this has helped me to synthesize my overarching questions for this project. The first being, how does TRPV3 lead to the pathophysiology of wood smoke or other toxicants? But before I could answer the question of what TRPV3 was doing in the lung epithelium after wood smoke particulate matter exposure, I first had to step back and ask the more basic question of what does TRPV3 do normally within the lung epithelium? And my hypothesis for this is that temporal changes in TRPV3 activity and expression after injury are essential for proper coordination of calcium homeostasis and the wound healing response, potentially through EGFR signaling and other growth signaling pathways. And so first I looked at the expression of TRPV3. So these are in our HBEC3KT cells, immortalized human bronchial epithelial cells. At a transcript level in A and B, I utilized different models of injury for A, and you can see as compared to the confluent non-injured control, TRPV3 expression is induced within 2 hours after pine injury, passaging, trypsinizing alone, as well as scratching these cells. And then over time, B is showing a time course of what TRPV3 transcript levels is doing dependent on confluence of these cells following passaging. So you can see on the left axis is TRPV3 transcript levels. On the right is the confluence of the cells. And TRPV3 is highly dynamic, cycles over time. And when the cells repair that injury and become confluent, TRPV3 expression is rather low. And then on the right, this is our Western blot showing TRPV3 at a protein level as compared to our TRPV3 overexpressors as a positive control. You can also see that TRPV3 cycles and is at different levels depending on the repair rate of these cells, which is what we would expect looking at the transcript profile of TRPV3. And then finally, utilizing a more functional assay, we looked at calcium flux that is specific to TRPV3 by using a TRPV3 specific agonist, drophonine, in a dose response. We looked at 2 different confluences of these HBEC3KT cells. In the white, these are cells that were 20% confluent, and then in the gray, these are cells that are completely repaired at 100% confluent. And you can see that calcium flux specific to TRPV3 is higher in the cells that are repairing. And so I was interested in modulating TRPV3 expression and seeing what that did to the repair process. So these are scratch assays where we grow cells to confluence, scratch them, and then measure the amount of time that it takes for these cells to repair the scratch wound. These are a different cell type. These are Bs2B cells, but they're also immortalized bronchial epithelial cells and are TRPV3 overexpressing Bs2Bs. And this scratch assay is quantified in A. You can see the TRPV3 overexpressing cells are a lot slower at repairing the scratch wound, and images from the scratch assay are represented in B. But I think the best way to look at these scratch assays is by looking at videos of them. So on the left-hand side are the normal Bs2B cells, and on the right are the TRPV3 overexpressing cells. And you can see how much slower they are. The normal cells tend to close around 6 hours, and the overexpressors, most of the time, they don't close the scratch at all. So we were interested in seeing what other factors were affected by TRPV3 overexpression. So we ran transcriptomic analysis on these overexpressors compared to normal Bs2B, which is represented at the top in A. This is an MA plot. And you can see TRPV3 in green is quite high because these are overexpressors, but also we notice that a lot of growth factors that dictate proliferation, wound repair, migration pathways were downregulated in these TRPV3 overexpressing cells compared to normal Bs2Bs. While the growth factors were downregulated, the receptors that they activate remained at a similar expression level. And we did confirm this with qPCR analysis in these 2 graphs here. There were some differences in TGF-beta 2 as well as TGF-beta receptor 2, which we have a few hypotheses on why this might be. involving adhesion and EMT pathways, but for the sake of time, I won't be going into that side of the project. I also looked at growth factors at a protein level. So on the right-hand side, this is an ELISA for HbEGF concentration. The B2Bs tended to have normal level or normal concentration of HbEGF growth factor in their media, whereas the TRPV3 overexpressors, it was non-detectable. So further emphasizing this idea that when you overexpress TRPV3, these cells tend to be slow growing and tend to have a downregulation of growth factors important for wound repair. And so knowing that, I wanted to see if I could partially rescue the slow-growing phenotype, so I supplemented some of these scratch assays of these TRPV3 overexpressing cells with some of these growth factors, and it turns out that you could boost the wound repair rate of these cells a little bit with when you add back in these growth factors that tend to be downregulated in these cells. And then furthermore, so this is switching back to our HBEC3KTs, I measured expression levels of these growth factors at an mRNA level, and they tended to all have a similar expression profile to TRPV3 expression following injury. And then also when we dosed these cells with these growth factors, it also increased TRPV3 expression significantly. And then downstream, I looked at pharmacologically inhibiting some of these EGFR receptor tyrosine kinases, and found that inhibiting most of them prevented TRPV3 expression following injury, but most likely it's EGFR or ERBB1 that is responsible for the most induction of TRPV3 following injury, and then some of the downstream components in the signaling cascade, including GSK3 beta, beta-catenin, and P38 MAP kinase. When I inhibited them, they all prevented TRPV3 induction following injury as well. And then finally, I was interested in other growth factor and signaling pathways. So I looked at Wnt and frizzled that system of signaling, as well as TGF-beta1 and TGF-beta2. And in summary, blocking components of these pathways also prevented TRPV3 induction. And then finally, looking at NF-kappa B, which is an important transcription factor, inhibiting this component as well prevented TRPV3 induction. And so I also looked at another form of modulating TRPV3 activity by utilizing TRPV3 specific antagonists. So all of these antagonists are structurally unique. Their structures are shown in A. And I also noticed that when I dosed these cells, these HBEC3KT cells, with these TRPV3 antagonists, and they were in a confluent monolayer, so no injury induction, these antagonists alone induced TRPV3 expression. But also when we ran scratch assays with these antagonists, they completely stopped or slowed the repair rate as well. So similar to the overexpressor phenotype of the slow repairing cells. And so in summary, I've shown you that when we injure lung epithelial cells, it tends to lead to an increase in TRPV3 expression. And over time, during the repair process, TRPV3 cycles and is dynamic and is dependent on how confluent the cells are. And we believe that this differential level of TRPV3 over time then affects calcium homeostasis to then affect the growth factor shedding and expression. And then those growth factors then affect the signaling pathways that they turn on. And then through my inhibitor data, I've shown that inhibiting these growth signaling pathways also prevents TRPV3 expression. So there seems to be some sort of feedback loop between TRPV3 expression and these growth factor signaling pathways. And then overall, TRPV3 overexpression and antagonism both prevent scratch wound closure and slow it down a lot. TRPV3 overexpressors tend to have a downregulation of these important growth factors, and TRPV3 antagonism also increases the expression of TRPV3, so that might explain why they have a similar repair phenotype to the overexpressors. And then secondly, I'll briefly go over some of my in vivo work that I've been doing recently with naphthalene. So naphthalene is cytotoxic specifically to club cells, which are non-ciliated epithelial cells. naphthalene is a PAH produced by combustion of any biomass, and so it is a component in both wood smoke and cigarette smoke. It has a strong mothball odor because it is the main ingredient in mothballs. Naphthalene requires bioactivation by cytochrome P450s to result in this epithelial cytotoxicity to club cells. And so I've included my dosing scheme on the bottom right here, where I'm utilizing TRPV3 knockout mice as well as wild type C57 black 6 mice, and then with one treatment of IP injection of either corn oil as a vehicle control or naphthalene at 200 mg per kg. And then we have been looking at a time course to measure endpoints such as airway mechanics and also pulling lungs for either histology or transcript analysis. And so my original hypothesis for this part of the project was that knocking out TRPV3 will hinder the epithelial cell repair rate necessary for complete resolution of naphthalene-induced injury in the mouse airways. However, this hypothesis has been changing a lot recently. But I will just briefly go over what I've been seeing in terms of histology slides. So this is an H&E stain. The top slides are from the airways of our wild type animals. The bottom side are TRPV3 deficient mice. On the left-hand side is the 12-hour corn oil control. The morphology of the airways of these animals at baseline, they're not too different. There are some slight differences that we haven't really fully characterized yet. But the more interesting thing is following naphthalene exposure, I don't know if you can see, but compared to the wild type, there is exfoliation and cytotoxicity to the epithelial cells in the airways, but the TRPV3 knockout mice tend to have a lot more exfoliation and cytotoxicity. And then in terms of resolution of repair, so this is 48 and 60 hours post-naphthalene exposure. Again, the wild type animals, their resolution of repair is fairly normal from what we see in the literature from researchers who use naphthalene. But with the TRPV3 knockout animals, it's striking how different the morphology is because it's highly disorganized. So they are eventually repairing, they're just not repairing in a normal way. So in conclusion, I've showed you that TRPV3 is activated by wood smoke particulate matter. It's also expressed within human and mouse lung epithelial cells. In vivo, wood smoke particulate matter results in remodeling of the airways, but that is also prevented when you block TRPV3 with a specific antagonist. In vitro, TRPV3 expression is induced within 2 hours and cycles over time dependent on the repair stage of the cells. And growth factors correlate in expression with TRPV3, but also induce TRPV3 expression. And inhibiting these signaling pathways that these growth factors activate during injury prevents the expression of TRPV3. Both overexpressing and antagonizing TRPV3 prevents wound repair, and our TRPV3 knockout mice, when they're treated with naphthalene, they exhibit distinct morphological differences, including a higher degree of exfoliation and a disorganized epithelium compared to the wild type animals. And then for the future, just really quickly, we do have TRPV3 knockout HBEC3KTs that I have been working on to characterize. This graph at the bottom is a scratch assay from looking at the knockout cell line to the normal HBEC3KTs, and you can see that it looks as though they repair a little bit faster than the normal 3KTs, which could be potentially interesting. And then with naphthalene treatment, we have a lot of animal studies planned for the near future to hopefully distinguish the extent of cytotoxicity and exfoliation that we see, but also the resolution of repair in this disorganized epithelium that we see in the TRPV3 knockout animals. And then finally, once we get a better idea of what TRPV3 deficiency is doing in this more basic model of injury with naphthalene, we can then go back to our pine wood smoke particulate matter exposure studies to determine the effect of TRPV3 knockout in those. And with that, I would like to thank the Riley Lab, especially Chris, and previous Riley Lab members, my dissertation committee members, The Venosa lab for teaching me really crucial surgery and necropsy techniques, as well as my funding sources, and a special thanks to the Skaggs Institute for Research for setting up this endowment, and to the Skaggs family for their ongoing support. And with that, I will take any questions.
Great talk. What is the family talk?
Yeah.
Curious if you sort of, or had a hypothesis teleologically, why would, what's the advantage of inducing TRPV3 after injury if the net result of it would inhibit repair?
Yeah. So the question is, what would the advantage of inducing TRPV3 be after injury? It's a good question for sure. It's something that's plagued me for a while, I think, through this project. But I think that part of that could be answered with the in vivo data that I showed early on in the presentation with our wood smoke particulate matter subacute exposure model, where we see that thick layer of epithelial cells after the subacute treatment of pine. I think that with that, the airways are almost protecting themselves by, so the activation of TRPV3 leads to the increased expression, but over and over again, it's almost beneficial because you're almost protecting your airways from future exposures, if that makes sense. Mm-hmm. Okay. No more questions? Thank you.
and chair of Andy Chibber's PhD committee. It's certainly my pleasure to introduce him. Andy came to us from the University of Kansas with a pharmacology background and an interest in outcomes research. So for a year, he took some classes to confirm whether that interest was real, and then he decided to dive into a PhD program. I think over the last 3.5 years, he has absolutely convinced himself of his interest and has gotten involved in a very exciting project that he's going to share a component of that with you all today. And again, also a very grateful recipient, as I am as well, of his Skaggs Fellowship grant. With that, Andy Chibber.
Sure, and can I remove the mask while I'm here? Or?
You can take it.
Okay. On this one? Okay, but it's inside. It's okay. Thank you so much, Diana, for the introduction, and thank you for joining in to listen to my presentation titled Economic Evaluation of Pharmacogenetic Testing Aimed at Identifying and Reducing Pulmonary Exacerbations in Cystic Fibrosis Patients. So here is the agenda for today in order to prepare you for what we would be discussing today. To begin with, I'll provide a brief background about the disease, which is cystic fibrosis, and pharmacogenetics, following which I will state the purpose and objectives of the research work. Moving forward, I will delve into the methods and results for the study, and in the end, I'll state major takeaway points of the research study to wrap up the presentation. I'll gladly answer any questions after that. So to begin with, cystic fibrosis is an autosomal recessive disorder that involves Cftr gene, which is called cystic fibrosis transmembrane conductance regulator gene. Now, this gene basically encodes a protein called the Cftr protein. Now, cystic fibrosis happens when a mutation in this gene happens, and which leads to basically misfolding of this Cftr protein. So as you can see here, because it's an autosomal recessive disease, you basically need 2 mutated copies of Cftr gene to have the disease. So if you have only 1 copy, you are called a carrier. If you have 2 copies, you have the disease. So let's look at what happens when you get a mutated copy or 2 mutated copies of Cftr gene. So in order to better understand that, we will have to look at the normal functioning of the Cftr gene or Cftr protein. So Cftr protein is basically a channel protein that basically pumps chloride ions into the secretions, as you can see in the diagram here. So this pump of chloride ions into the secretion basically pulls water from outside of these secretions, thinning these secretions, making them viable. But when a mutation basically in Cftr gene occurs, for example, delta F508 mutation, this protein basically misfolds. So there's no more protein there to regulate that a movement of chloride ions, which basically makes these secretions very thick. So they are not thin anymore, they're thick. So these thick secretions can lead to a number of issues. And I'm going to discuss those issues in the next slide. So for instance, in early childhood, these thick secretions can lead to pancreatic insufficiency, leading to poor weight gain, failure to thrive. After a few time, due to these secretions, the pancreas get damaged, which can lead to acute pancreatitis, and after fewer episodes, it's actually lead to chronic pancreatitis. Eventually, after a lot of damage to pancreas, the endocrine function of the pancreas is lost, and unfortunately, these CF patients basically have insulin-dependent diabetes at that time point. So lung-related problems, which is cystic fibrosis is usually known for, usually occur later in the childhood. And these lung-related problems happen because of the thickening of the mucus, which happens in lung. And this thickening of the mucus basically leads to repeated CF exacerbations in these CF patients. And eventually, after some years, even some decades, this leads to respiratory failure, which is leading cause of death in the CF patients. Now, the treatment, unfortunately, CF has no cure. We can only manage this disease. So treatment actually comprises utilization of several therapies. For example, antibiotics that is used to manage the infections caused by those thick secretions. Pancreatic enzymes, because as I've already mentioned, the endocrine function of the pancreas are basically lost. Then there are medications such as mucus thinners that basically work to thin the mucus to get that mucus flowing somehow. And then there are breakthrough therapies such as Cftr modulators, as you can see here, that work by targeting specific Cftr mutation types. An example would be a combination of lumacaftor, ivacaftor. They work together to improve the dysfunctioning Cftr protein, basically. So thanks to these therapies, an average CF patient now can live a better, a healthier life compared to I would say like 2-3 decades ago. For instance, 49.7% of CF patients got to be 18 years or older in 2013 compared to a mere 29.2% back in 1986. In addition, the median age of these patients is well into 40s now, thanks to these therapies. So these therapies are helping these patients, but now we are running into a different kind of problem. So as you can see, these patients are on so many drugs. They are living healthier life compared to a few decades back. But the problem that we are running into now is basically how to manage these kind of medications or these convoluted regimens. So for instance, an average CF patient can spend more than 2 hours just managing or just focusing on their treatments in a day. So that translates to about 1 month in a year just focusing on your medications, just taking your medications, which is a significant amount of time. In addition, the median number of daily medications in CF adult patients has risen to 7, resulting in treatment complexity and increased risk of adverse drug reactions and subsequent interference with the overall treatment goals. So we are thankful for these therapies that are making them live longer, but we are running into these adverse drug reaction issues now because they are on so many drugs. So these adverse drug reactions are the focus of my dissertation work, a portion of which will I be discussing today. So these ADRs are very substantially among patients depending upon two factors. The first factor is the medication profile of the patients, and the second factor is the patient genetics. So for instance, genetic polymorphisms of CYP450 gene or drug interactions related to the metabolism of CYP450 enzymes can lead to variable treatment response and ADRs in these CF patients. So the idea is to basically find an intervention that can use both medication profile of the CF patients and the genetic profile of the CF patients to guide a pharmacotherapy option, and basically hopefully help us reduce the significant proportion of ADRs that these CF patients are facing. And that's exactly where the personalized medicine comes into the place. So personalized medicine is an emerging field focused on utilizing individual-specific factors such as genetic risk profiles and to explain and basically predict patient responses to different therapies. So, so what's happened, so as you can see in the figure, there's a classical medicine and there's personalized medicine. In classical medicine, we take the trial and error method because there's a bunch of people that have different individual level factors, and medications sometimes don't work for certain kind of patients because they have different genetic factors. So precision medicine basically uses a succession of decision rules which map up to-date patient information and clinical evidence to recommend action for pharmacotherapy. So as I mentioned, this negates basically the usage of trial and method error and basically can help us increase or better the health outcomes in CF patients. So modern tools of precision medicine, there's pharmacogenetic panels that we can basically use as an intervention in these patients. And these panels can help us identify genetic variations associated with the drug response based on the individual factors of these patients. So these tools or pharmacogenetic panels are not new. They have been around for quite some time. They have been used in disease areas of oncology, cardiovascular diseases, but our research is trying to see whether using such a pharmacogenetic panel in cystic fibrosis would be clinically viable or economically viable. So, and that's exactly what we did in another part of my dissertation work. So we basically took two groups of patients, one patients that were on standard care. So we only utilized medication profile data of these patients, and we compared them to a PGX-assisted care arm. And the good thing about this arm is that the medication profile level data was supplemented with genotypic level data of these patients. And we found that the inclusion of pharmacogenetic data can help clinicians predict and reduce number of ADRs in CF patients. So in this figure, you can see that 88% of the patient populations, 50 some odd patients, can have better health outcomes if they undergo a pharmacogenetic test, basically. We're not going to go into the details for the results of this part, but this is the first part of the puzzle. So I'm going to summarize everything that we have gone through till now. So we have a disease, cystic fibrosis, very debilitating disease. We don't have a cure, but we have therapy options that can manage this disease pretty well, or we are improving in managing this disease, but we are running into problems of adverse drug reactions and treatment complexity. But we have identified an intervention in the form of pharmacogenetic panel that can help basically reduce these ADRs in cystic fibrosis patients. So clinically, we see that this works in CF patients. 88% is a pretty significant number. The second piece of the puzzle, and what we're going to talk about today, is, okay, clinically it makes sense to do this kind of pharmacogenetic panel in CF patients, but whether financially or economically it would make sense to do this kind of panel in the CF patients. And the rationale is sometimes these pharmacogenetic panels can be sometimes so costly that the health improvement that you see in a certain patient population does not warrant the use of expensive pharmacogenetic tests per se. So the idea is to basically see whether the cost effectiveness of a PGX panel aimed at preventing reducing ADRs in pulmonary CF patients is viable or not. So to realize this aim, a Markov state transition model was developed, looking at patients heterozygous for a certain mutation. You don't have to worry about the mutation, but this mutation is the most common mutation found in the CF patients. So the time horizon for this model was 80 years, and with a 1 month cycle, that means the model keeps going. We put in probabilities after for every 1 month. So after 1 month, a patient can transition basically from one transition state to another transition state. On top of 80 years, we did a scenario analysis looking at 2 years, 4 years, 8 years, 10 years, 20 years, and 40 years worth of time horizon to look at the short-term benefits and long-term benefits of doing a pharmacogenetic panel in these patients. So again, I'm going to reiterate the comparator arms. There's a standard care arm, and this arm of the comparison entails the usual care of the CF patients, including the number of drugs. The pharmacogenetic testing assisted care arm, and this arm basically mimics the standard care arm, but the treatment care is guided by a preemptive PGX testing that is aimed at reducing ADRs in these CF patients. For the outcomes, we are looking at costs, we are looking at QALYs, which is quality adjusted life years, and rate of pulmonary exacerbations, which is a kind of ADRs in these CF patients. So this slide is basically going to show you the health state transitions possible for a CF patient. So the idea is, people who are not familiar with this, the idea is that every 1 month, a patient can move between these health transition states. So we simulated this model for 1,000 patients. So all 1,000 patients basically started in the well state. So they will be in the well state until a probability arises that they're going to move either to a minor, moderate pulmonary exacerbation, or they can move to a severe pulmonary exacerbation. They're going to be there until once they reach that. So the classification of minor, moderate pulmonary exacerbation and severe pulmonary exacerbation, they are classified based on the kind of treatment these CF patients receive. So a pulmonary exacerbation was deemed to be a minor, moderate pulmonary exacerbation if they only received oral antibiotics for the treatment. If a patient has to stay, if a patient has an inpatient stay, that pulmonary exacerbations was classified as a severe pulmonary exacerbation. So after reaching either of these stages, they can either go back to well stage, as you can see in the figure, after getting the treatment, or they can die from the pulmonary exacerbation. On top of that, a CF patient can just move from well state to natural death state, and that death has nothing to do with pulmonary exacerbations. So that's just death due to natural causes. So impact inventory. So this slide is going to show the boundaries of our analysis. Cost effectiveness analysis can be done in a multitude of ways. They can capture direct cost, indirect cost, social cost. But our analysis is very straightforward. We basically focused on the payer and healthcare perspective, and only direct costs for the patients were basically captured in this analysis. So any kind of non-medical direct cost, indirect costs, savings that can occur from doing such a pharmacogenetic test was not captured in this analysis. So this table basically shows the key model inputs that were utilized for the simulations in the model. And you don't have to basically focus on anything, but the main idea is that these inputs are derived from a systematic literature review that was conducted. And the inputs were then run by a clinician, a pharmacist, and an economic modeling expert for internal validity. So, which is pretty standard for a cost effectiveness analysis. So coming to the results, this first slide is going to show, so we ran the simulation, we ran the model. So this first slide is going to show you the health outcomes and healthcare cost between these two comparator arms. So on the left side, you can see the health outcomes, which is basically QALYs, quality associated life years. And you can see on the right side, you can see the cost, healthcare costs associated with each comparator arm. You can see that PGX assisted care generates more QALYs, as listed here, 17 versus 16, but it actually leads to less healthcare costs because we are preventing more pulmonary exacerbations in this patient group when compared to standard care. So all in all, just looking at the health outcomes and costs, looks like that PGX assisted care is better than the standard care here. So this slide is going to show the trace plots for the standard care versus PGX assisted care. people who are not familiarized with the trace plot, it's pretty simple. Trace plots basically follow the patients throughout the whole time horizon of the model, that is 80 years. And basically at each state, it's going to show the probability of a patient being in a certain transition state. So it will be more clear once you look at it. So the top one is the standard care. The bottom one is the PGX assisted care group. Now, if you look closely, you can immediately tell the differences between the two trace plots. Three major takeaway points from this figure. First of all, there are more people in the well transition state compared in PGX assisted care group compared to standard care. Second thing is that less people are having severe PXs, pulmonary exacerbations, and minor moderate pulmonary exacerbations in PGX assisted care group compared to the standard care group. And also, not only they are getting less PXs, the pulmonary exacerbation deaths are significantly reduced in the PGX assisted care group compared to the standard group here. And because they are not dying from the PGX death, they are dying from the natural causes, and that's why you can see the purple area here is bigger in the PGX assisted care group when compared to the standard care group. So this slide is going to talk about the ICER for the standard care versus PGX assisted care. So concept of ICER, again, who's not familiar with the ICER, it's called incremental cost effectiveness ratio, and basically it's looking at the difference of costs of how many our two interventions, let's say, divided by the effect, or let's say QALYs in this case, for two interventions. So basically, it's looking at cost per effect. And you want your ICER to be lower because you want to pay less for, let's say, a QALY gain in order to be cost effective at a given willingness to pay threshold. So all this slide here and table here is showing that PGX assisted care, again, generates more QALYs and has less cost related to healthcare. And that's why the standard arm was strictly dominated by PGX assisted care arm, as you can see in the figure. So it's just strictly better than the standard care arm. Now this ICER is actually utilized sometimes as a decision-making tool. There's pros and cons, but yeah, it can be sometimes used as to disperse healthcare resources. So good to see that our PGX assisted care arm is doing better, has a lower ICER, let's say, compared to the standard care arm. So in order to, now I showed you a table which had a ton of inputs in it for the model. Now these inputs come with issues of uncertainty, variability, right? Because you pick these inputs from the literature, but you have a point estimate, then you have a range, and then you have to basically consider the variability for your point estimate. So to consider the point variability within the point estimates, we did a sensitivity analysis, which is called a deterministic sensitivity analysis. And this table basically shows you that the two factors that can influence the base case results that I've shown you is the cost of, so basically the input, the parameter estimate we got for the cost of severe PEX and the probability of getting the severe PEX. So the second type of sensitivity analysis that we did was probabilistic sensitivity analysis, and this has more to do with the uncertainty in the parameters. So what we basically do is we run a number of simulations over and over again, and we see how the results change in each simulations, because it's gonna pick the estimate from a distribution that was assigned to it. So in here we ran 10,000 simulations back to back for the same thing, and we found that in 64.8% of the simulations that we ran, of those 10,000 simulations that we ran, the PGX assisted care arm was preferred over the standard care arm, which basically implies the results are pretty reliable as far as this dominance is concerned. This slide is going to show the cost effectiveness acceptability curve for standard care versus pharmacogenetic guided care. I told you about the ICER, but there's a thing called WTP, and this is willingness to pay threshold. So this is basically the amount a payer is willing to pay for a certain intervention. So the probability of an intervention being cost effective can change depending on the willingness to pay threshold. For example, if I'm willing to pay $30,000 for an intervention and ICER is actually higher than that, then I'm not going to pay for it. But if its willingness to pay is, let's say, $150,000 and ICER is lower than that, then payer would obviously pay for it or would be willing to pay for it. So all this figure is showing here is that the probability of PGX-assisted care being cost-effective compared to standard care was 0.55 or higher at any given willingness to pay threshold. So it doesn't matter what kind of willingness to pay threshold there was, our PGX-assisted care arm was better than the standard care arm. So as I mentioned before, on the top of the 80-year analysis, we did a scenario analysis where we changed the cycle of our model. So we did 2 years, 4 years, 8 years, 10 years, 20 years, and 40 years. And basically on the right, you can see that irrespective of the length of the time period, the standard care got dominated by PGX-assisted care. So you can see increased QALYs as soon as like 2 years after doing a PGX, which is pretty significant. So for the conclusions, so to conclude everything, so polypharmacy and resultant ADRs have significant economic implications. Intervention to reduce these ADRs are likely to be cost-effective in patient populations such as cystic fibrosis. As you have seen, a small increment in the utility value, so there was not a lot of difference between the QALY values, but there was a huge cost benefits associated with that small QALY increase. Now, PGX-assisted care in CF patients can lead to better health outcomes and economic outcomes, as demonstrated. And future research should evaluate which CF patient subgroups are most sensitive to such an intervention, or there are some patient groups that might not benefit from such an intervention per se. Another important point that I would like to make here in the end is that the results should not deter providers from prescribing whatever medications they have to, because in the end we want better health outcomes for CF patients. But if those decisions can be supplemented by, let's say, a PGX panel, I think that would lead to better health outcomes in CF patients in general. So I think that marks the end of my presentation. I'll take any questions. Thank you. Yes.
So, and I maybe I'm just overlooking what you said. So it looks like the major driver of the difference is the ability to the number of serious events, like the cost and so on. Like kind of going back to the premise for you is the idea that the pharmacogenetic testing will address average drug events. But how much of the shift, the transition of a patient from a mild to a severe pulmonary occurrence is driven by average drug reactions or those kinds of things that you're looking at versus or an inadequate treatment for the disease state and therefore they transition. And so I guess I'm not seeing quite the link between the idea that we have all this polypharmacy and it can lead to these average drug reactions, which is quality of life. And so we want to test, but I'm not clear about how we're trying to test things to ensure enough to be a treatment that that's the transition to
So that's the second part of the dissertation that I briefly touched upon, but the input is going to come from that part. So we are still in the analysis part of that phase, and that input is basically going to tell us if we do this kind of PGX test, how much adverse events it's going to basically reduce. And that probability and that percentage is basically going to, so we're going to revise this a little bit once we get that input. And this work is ongoing right now, but I think the assumptions that we made here are pretty broad. So I think the base input that we took was like 20%, and then 25% plus minus of that 20% was basically used to model this. So I was pretty restrictive of that percentage amount. I think we're gonna probably see like 30% or like 40% when we get the results from the second part of the study. So pretty restrictive, and that's why we are probably not seeing that much quality gain. But I'm pretty hopeful that it's going to be in the same direction when we get those results.
I have a similar question about thank you for answering that. It's a great talk. The question I love was, so when you're talking pharmacogenetic assisted therapy, are you talking about specific testing, or are you talking about looking at the entire panel of tests that might inform the wide range of drugs that we use?
So the second option, so basically for the patients, for the second part, we did the whole CYP450 enzyme panel. So it's actually a panel of all the CYP450 enzymes. Yeah.
So then, the follow-up question then is, how will it impact your analysis if you do look for a specific event, i.e., 384, we know, plays a role in the metabolism of the Cftr modulating drug.
Yeah, that.
The specific fit, that improves your cost benefit with pharmacogenetic?
So it's two jumps, right? So right now, the pilot study that we did, the second study, it's for the whole panel. So we're looking at whole, but I think a larger study might be needed if we want to do something like a very polymorphism specific mutation, right? And then a model would have to be made just based on that, on basically the clinical output that we're going to see from that study. So I'm not sure whether we're going to see any kind of effect if we just focus on one, let's say, right? But I don't know. I think it might have to be a bigger study, first of all, clinically, a trial, prospective trial, for example, and then a CE has to be based on the findings of that trial to get to that answer. So, but this is pretty new in cystic fibrosis patients right now. Like, people don't do this. So it's just the beginning. It's just to start the discussion right now.
So you mentioned earlier that there is a cure for CF. Has there been any progress towards gene therapy?
There have been, but it's not as big as something like CFTR modulators right now. So it's in pipeline. So there might be cure, but in decade or so, I don't know. Yeah, but right now there's nothing. Yeah. Mm-hmm. Please. Sorry? Part of it.
Good afternoon. Thank you. Good afternoon. I'm Hamid Gandhari. I have the privilege of being the advisor for Nitish Khurana. Nitish joined us from Pune University, where he did his master's in medicinal chemistry, and prior to that, he did his honors in chemistry from Pune University. During his PhD studies, Nitish worked on a unique project to try to characterize the sinonasal vasculature in order to deliver drugs more effectively and less toxic, with a less toxic manner, to the sinonasal cavity for many people that suffer from chronic Rhino sinusitis. This project was actually co-supervised with a physician scientist, Dr. Jeremiah Alt, in otolaryngology. Nitish.
Thank you, Dr. Gandhari. As he said, the title of my talk today is Vascular Pathophysiology Characterization in Chronic Rhinosinusitis for Controlled Drug Delivery. So even though for the last couple of years I've practiced every day to say chronic rhinosinusitis quickly 5 times every day, it still sometimes twists your tongue. So from here on, I'll refer to chronic rhinosinusitis as CRS. So let me first introduce what CRS is. Chronic rhinosinusitis is a debilitating inflammatory disorder of the sinonasal mucosa that affects the paranasal sinuses, and it affects up to 12-15% of the US population. Patients with CRS experience significant decline in their quality of life, as is stated by the state metrics, and their patients often experience a lot of inflammation in their mucous membranes, where there's a lot of production of mucus in the sinuses. In US alone, in a year, we spend about $64 billion towards CRS, which includes both the treatment and the productivity costs, which amounts to about 3 to 5% of the US annual healthcare budget. So here you can see the anatomy of the sinuses in humans. So we have 4 different sinuses that are separated on both sides of our face if we divide our face sagittally along the septum. The first is the maxillary sinus, which is in the maxillary bone behind your nose. Then you have the ethmoid sinus, which is in the orbit. You have the sphenoid sinus, which is the most posterior sinus located under the sphenoid bone. And then you have the frontal sinus, which is behind your eyebrows and is the most anterior sinus in our face. So what makes chronic rhinosinusitis chronic? That's based on how long does the symptoms prevail for in patients. If the symptoms prevail in the patients for up to 4 weeks, then it's called acute rhinosinusitis. If they prevail for about between 4 to 12 weeks, then it's called subacute. And if they prevail for more than 12 weeks, that's when it's called chronic rhinosinusitis. Some of the symptoms that the patients go through are thick nasal mucus, plugged nose, headaches, hyposmia, nasal discharge, and facial pain. Now, a patient is diagnosed with CRS when he or she suffers through or goes through 2 or more of these symptoms for about 12 weeks, along with inflammation for 12 weeks or discharge of mucus from the nasal passage. Now, CRS is divided into 2 basic phenotypes. One is CRS with nasal polyps, and the other is CRS without nasal polyps. So nasal polyps are these benign water-shaped sacs that are formed from the upper lining of the septum in our nose, but they're benign in nature. So the patients between these 2 different phenotypes often experience a different inflammatory response, or they have different symptoms at times. Now let's look at some challenges in CRS and what are the unmet needs. So some of the challenges in CRS, as I said, is that the patients experience a lot of decline in their quality of life. They develop comorbidities such as migraines, cognitive depression, and sleep dysfunction. CRS as a disease is more prevalent than diabetes, and more antibiotics are prescribed in US for CRS compared to any other disease. And the quality of the life, as decided by the state health metrics for CRS, is as bad as other diseases like coronary artery disease or Parkinson's. So here you can see a CT scan of a patient who has CRS. This is the maxillary sinus, and this is the ethmoid sinus. As you can see, that there is a lot of opacification in the airways of this patient, which means that most of this, the airway in the sinuses in this patient is filled with very thick mucus. Now, what are some challenges in the medical management? So currently, all the therapeutic management that is available for CRS right now is to relieve the patients of the rhinological symptoms, which includes a combination of saline irrigation, which is usually given in adjunct with other treatment options, such as topical and oral corticosteroids and topical or oral antibiotics. Now, the problem with topical corticosteroids is that the turnover time of mucus in the sinuses is so quick that the residence time of the drug inside the sinuses becomes really low. Also, as I said, that there is presence of very thick mucus in the sinuses, so the penetration power for these drugs is really low, and because of that, the distribution of the drugs across the sinuses, where it has to reach the inflamed tissue, is only 3 to 5% of the administered dose. So oral corticosteroids are effective, but the problem with oral corticosteroids is that they present with a lot of toxic effects and a lot of systemic side effects. Antibiotics are also effective, but the issue with antibiotics is that patients often form resistance against these. Now, for the last 15 years, all research has been focused on topical corticosteroids because topical corticosteroids is one of the most commonly prescribed treatment options for these patients. So because of that, the research has been only focused on topical corticosteroids, and nobody has tried to circumvent the current available therapies to find better treatment options. Because of that, more than 20% of these patients turn to surgical management, because of which more than 500,000 endoscopic sinus surgeries are performed annually in US. There is a problem, however, with surgical management is that there are a lot of surgical risks, such as cerebrospinal fluid leakage, intraoperative hemorrhage, and osteostenosis. Not only this, there's always risk of damaging the eyes because the eyes or the orbit of the eyes are very closely located to the frontal and the ethmoid sinus. And this is not the only issue. More than 50% of the patients often have recurrence of this disease. So because of this recurrence, they have to again go back to medical management, which doesn't often work for these patients, and then again have to return to the surgical management in a span of 10 to 12 years, which becomes a really vicious cycle for these patients. Because of this vicious cycle that they have to keep circling between medical management and surgical management, it's not only a socioeconomic burden for the individual, but also for the society. So we wanted to now circumvent the currently available therapeutics for CRS and see some other options that could be beneficial for treating these patients. So we know that for other inflammatory diseases, such as atherosclerosis, rheumatoid arthritis, inflammatory bowel disease, vascular permeability has been used to deliver drugs or nanocarrier systems systemically via systemic injections. And this happens through a mechanism called ELVIS, where macromolecules can extravasate through these leaky vasculatures that are present in these vascular dysregulated vasculature. These macromolecules can extravasate and accumulate in the inflamed disease site. This effect is very similar to EPR effect or the enhanced permeability and retention effect like we see in tumors, where similarly macromolecules or nanocarriers can extravasate through these dysregulated fenestrations and accumulate in the tumors. So similarly, we thought that if we go the systemic route and use a nanocarrier system and inject that via IV or via the systemic administration route, the macromolecule will extravasate through these dysregulated fenestrations or interendothelial tight junction gaps, and they will accumulate in the inflamed sinonasal tissue where they can release the payload. Now the question is, we don't know whether the vasculature is dysregulated in CRS patients or not. So first what we wanted to do was to establish this vascular dysregulation and whether we can exploit this vascular permeability to deliver macromolecules in these patients. So first we ran a meta-analysis and we observed that there was upregulation of vascular signaling pathways such as vasculature development and blood vessel morphogenesis. And we also saw a significant differentially expression of vascular-related genes such as PCAM1 or EGR3. Now, this was an indication that, okay, we are on the right path. We are seeing some kind of vascular dysregulation in CRS patients compared to controls. So next, we actually collected sinus tissue samples from human patients, both the CRS patients and controls. As you can see here, we first analyzed the fenestration sizes in the blood vessels present in the sinus tissue. Here you see a fenestration size of up to 60 nanometers in a CRS with nasal polyp patients, compared to a control patient where the fenestration size does not increase 12 nanometers in size. So we did see an increase in and a common trend where CRS patients had bigger fenestrations or interendothelial tight junctions compared to controls. And in some cases, we even saw fenestrations of up to 140 nanometer in size, which suggests to us that there is increase in vascular permeability in the form of increase in interendothelial tight junctions. Next, we wanted to look at the blood vessel morphology. So as you can see here in this control patient, all the three layers of blood vessel, which is the tunica intima, which is the most internal layer, then the tunica media, which is the medieval layer, and then the tunica extrema, all three layers are intact in this in the blood vessel of this control patient. Whereas for both CRS without nasal polyp patients and CRS with nasal polyp patients, we see a loss of the outer two layers of the blood vessel. We also see fractures in the basement membrane. And not just fractures in basement membrane, if you compare the thickness of the layers here, we also see a global thinning in the luminal and the abluminal lining of the blood vessels. So this confirms that there is an increase in blood vessel morphogenesis, which also corroborates the story which we observed in the meta-analysis, where we observed an increase in the signaling pathway, which was blood vessel morphogenesis. Further, we also wanted to evaluate the number of blood vessels and compare that between control and CRS patients. So as you can see here, we performed PCAM1 immunohistochemistry, and PCAM1 is a biomarker for endothelial cells. So as you can see here in the quantification, we saw a significant increase in the number of blood vessels in both CRS without and with nasal polyp patients compared to controls. Now what this suggests is that we're observing an increase in neoangiogenesis and neovascularization, which is as expected because inflammation and angiogenesis are cause and consequence of each other. We know that for new blood vessels to grow, there is a constant need of supply of water, nutrients, and oxygen. And for that supply to happen, these tight junctions tend to form larger gaps between them, which causes this vascular permeability. So here we're observing an increase in number of blood vessels in terms of the quantification, which reflects an increase in angiogenesis. So when all these results are put together, what we observe is that the inflammation drives the vascular dysregulation. So we see an increase in neoangiogenesis and neovascularization. We see an increase in blood vessel morphogenesis, and we see an increase in vascular permeability in the form of increase in interendothelial tight junction gaps. Now we have proof that we do see vascular dysregulation in these patients and this type of inflammation. So we wanted to take advantage of this for delivering systemic therapeutic nanoscale drugs. So based on that, we came up with the hypothesis. We know that the current therapies like we talked about, which are the nasal sprays and the oral drugs, are not very effective right now. So we want to circumvent these current therapies, which was the key hypothesis of this project. But the final hypothesis of this project was that we want to take advantage of this blood vessel dysregulation and permeability to deliver nanoscale carriers where they can extravasate and accumulate at the diseased inflamed site. And recently this article was selected as the feature article for the Nanomedicine Journal. So quickly I want to discuss the model nanocarrier system that we selected and also the animal model that we work with to evaluate the accumulation of these nanocarrier systems, whether the systemic route works or not. So the nanocarrier system that we selected was a liposome structure. We know that liposomes have a low toxicity profile. They have the ability to incorporate both hydrophilic and hydrophobic drugs. They can protect the drugs from activation in the bloodstream, and it allows multifunctionality where we can incorporate different entities such as imaging agents on the surface. We can incorporate different targeting antibodies on the surface. We can incorporate different radioisotope chelators either in the core or the surface. So it allows multifunctionality where we can use it for many different purposes. The imaging agent that we selected was indocyanine green or ICG. ICG is a near infrared dye which is FDA approved. But ICG is unstable at physiological temperatures for more than 24 hours, as it tends to fragment and agglomerate. So here we show the DLS characterization and the TEM characterization of the liposomes that we synthesized. So I've selected two different sizes of liposomes. One was 140 nanometer in size, the other was 70 nanometer. and then empty liposome was used as a negative control. As you can see that we got approximately 140 and 70 when we characterized the size using dynamic light scattering, and we saw similar results such as 132 and 78 when we characterized the results using TEM. So we do have stable and uniform liposome formulations with low polydispersity as well. So now quickly I want to talk about the mouse model that we use. So this mouse model is an allergic CRS mouse model, and it has translational properties. The best aspect of this mouse model is that it mimics the inflammatory response in the animals as we see in humans, both in terms of the inflammation, looking in terms of the inflammatory cytokines, and looking in terms of the vascular pathophysiology. Now, previously the vascular pathophysiology was not established for this animal model. So it became important for us as well to establish the vascular pathophysiology so that we could compare it with the human vascular pathophysiology that we were looking at, and we wanted to see whether the vasculature in this animal model is dysregulated as well, and can we take advantage of that to deliver nanoscale carriers. So how this model is developed is that we use Aspergillus fumigatus, which is a fungal extract, and we inoculate biopsy animals intranasally with this Aspergillus fumigatus 3 times a week for a continuous of 8 weeks. We do that continuously for 8 weeks so that we see a chronic response and a chronic inflammation in the sinuses of these animals. And then we performed imaging using in vitro imaging system for about 21 days, and at the 21 day, we euthanize these animals and collect tissues for different downstream assays. So here quickly I'm showing the study design. So we had different study groups, which were the pegylated liposome with the indocyanine green, which is the imaging agent at 70 nanometer in size. Then the liposome with ICG at 140 nanometers. Then we have the free ICG, which is just the dye by itself, and we have the empty liposome, which is the negative control. And then we have 2 different animal groups. One is the diseased animal, which is the allergic CRS mice. And then we also have the control, which were the healthy animals, which were the animals that were just in the vivarium for 8 weeks. So first, as I said, that we wanted to confirm whether we observe vascular permeability and vascular dysregulation in the diseased animals compared to controls. So as you can see here, we demonstrated that we see an increase in fenestration sizes in diseased animals compared to controls. So here you can see that a fenestration of 140 nanometer was observed in CRS mice, whereas for control animals, it did not exceed 10 to 11 nanometers, which was very similar to what we observed in humans as well. In some cases, the fenestrations for the allergic CRS animals went as high as 500 nanometers. And in quantification, you can see that we saw a significant increase in the fenestrations in diseased animals and in controlled animals. We also observed similar results in terms of changes in blood vessel morphology, where we saw fractures in the basement membrane. We also saw a thinning, global thinning of the luminal and the abluminal endothelial barrier thickness, but I'm not showing, those results were significant, but I'm not showing those results here. So when these, then we performed iris imaging on these animals to look at the accumulation and retention of these liposomes in the sinuses. So as you can see here, for the CRS group, we saw a significantly more accumulation of the pegylated liposome at 140 nanometer compared to the control. In the figure, you might think that we see a similar accumulation, but if you come here to the quantification part, we do see a significant increase in the accumulation of these liposomes in diseased animals versus controlled animals throughout the 21-day study. For the 70 nanometer, we do see a trend where we see an increase in accumulation of these liposomes in diseased animals versus control animals. And at the last day of the study, we also observed a significant increase in the accumulation of these liposomes, the smaller liposomes in the diseased animals compared to the control animals. For free ICG, as you can see here, that after 24 hours, we see no signal coming from free ICG, because as I said, that free ICG is not stable at physiological temperatures after 24 hours as it tends to fragment and agglomerate. Now, finally, we wanted to see that what's the exact location of the accumulation of these liposomes based on the anatomy of the mouse sinus. So as you can see, the mouse sinus has three different, well, the mouse anatomy has three different sinuses. One is the ethmoid, one is the nasoturbinate, which is on top here, and then one is the maxilloturbinate, which is at this portion. Now, we saw that most of the liposomes were accumulated in the ethmoid sinus, which is as expected, because when we generate this animal model, we see that most of the inflammation in these animals is also observed in the ethmoid sinus and not in the nasal or the maxilloturbinate. So it was expected that the liposomes should accumulate in the ethmoid sinus because that's where the vasculature will be most dysregulated. Finally, we also, while performing TEM imaging, observed intact liposomes in the sinuses, which were present in the ethmoid sinus of these animals. So as a conclusion, liposomes were accumulated and retained in the sinuses for up to 21 days. So quickly in summary, I want to say that we saw an increase in vascular dysregulation in disease versus control, both in human sinus tissue samples and mouse sinus tissue samples. We saw an increase in fenestration sizes in both humans and animal sinus tissue, tissue sinus in both disease compared to control. And then we also observed accumulation and retention of liposomal indocyanine green in the mice for more than 21 days. And we can now afford controlled drug delivery, which can be helpful, and it can be helpful in treating and circumventing the currently available therapeutic options. And this project has also set up a stage for investigating further arms of the project, where different students in the lab are working on different arms of this project. Some of the future directions is, so we know that this preclinical study will set stage for a clinical investigation because we show translational capabilities as well. We want to correlate the extent of accumulation with the extent of inflammation, because in humans, the patients who suffer from CRS, the extent of inflammation can be very heterogeneous, where even they don't have symptoms, there can be acute or subacute flares in these patients. So it would be essential to correlate the extent of inflammation with the extent of accumulation in these animal model and then in future in humans as well. We also want to investigate the efficacy and toxicity by incorporating different inflammatory drugs such as corticosteroids or such as budesonide in liposomes and look at the efficacy and tox. And this project also helped in setting the stage for a future NIH R01 grant. With that, I would like to thank my mentors, Dr. Gandhari and Dr. Alt, for their constant guidance and support. Thank you to the Skaggs family and the Skaggs Foundation for giving me this opportunity to finish this project in the last year. All the lab members, all the collaborators, all the patients that were involved, and all the animals as well. And finally, all the funding opportunities, the Skaggs Research Fellowship, the SOM Seed Grant, the NTP Fellowship, and the Head and Neck Seed Grant as well. Thank you. So we don't have a control over the release of the drug from the liposome. And right now, as you see, currently we have just used an imaging agent. The next step is to actually incorporate a drug in there. There are ways in which you can control the release, but as of now, we're just hoping that there will be a sustained release of the payload from the liposome. Right. So there are different underlying causes for CRS. So like one of them would be formation of biofilms. And last week, a student of our lab, Bhuvan, talked about that. So there can be like accumulation of a lot of bacteria, fungi, viral stuff in the mucus because there is formation of biofilms. Because of that, it becomes really difficult to treat that. And there's just a constant. So basically, you can't stop the discharge of mucus, right? Essentially, once the production is happening, you can clean the mucus after every few years or so, but it's difficult to control that aspect. You can treat the inflammation, but you can't stop the symptoms arising from the other aspects of CRS. And that's the issue because the current treatment options that are available do respond to the inflammation, but they also either present with a lot of toxicities or there's just very low penetration power of the drugs, and it's not very effective at the moment. No further questions? Thank you.